Hey, this is Coach Boyson. In this screencast, we're going to be talking about mutations. When I think mutations, I think X-Men. You know, this pose right here, the old Wolverine. But mutations, just like in that movie, obviously that's science fiction, but they're changes that happen to the physical characteristics because of a mutation that happened at the DNA level. And so obviously if you watch that movie, you're going to see all kinds of different characteristics of these, or what they call powers maybe, because these physical changes that are occurring because of a mutation that happened at the DNA level. And so obviously not real life there, but just an example to kind of get you in the right frame of mind. Here's another picture. Not really a mutation, but I thought it'd be really, really cool to see something like that. And so a couple mutations that actually are real. Most mutations, so you know, are not good things. Most mutations that happen aren't beneficial. And so it's not something where there was this movie that came out a long time ago called Waterworld. Really bad movie, Kevin Costner. But anyways, the world's covered in water, and so he was born and had gills, was able to swim underwater. Mutation, right? That was beneficial to him. Most mutations are not beneficial, like in this case here. And most mutations, a lot of them do happen to our limbs, our hands and feet. I had a friend in high school that had webbed toes, webbing between his toes of skin. Here you can see this person, if you count the fingers, has 6 fingers. On each hand, and a couple of them actually have to kind of be growing together there. And so that's an example of a mutation. There's a change in the DNA that caused this weird the proteins to change, which caused this mutation to happen. Here's another mutation. We have a snake with 2 heads. Again, not a great mutation. Maybe it is, maybe it isn't. That's probably not a good mutation for this animal. Otherwise, we'd see a lot of snakes with 2 heads. And so most of them aren't beneficial. I do want to give you an example of one that might be beneficial out of this little cartoon picture. You can see here the bird on the right's able to reach the food or reach the insects. And so going from the beak on the left and then this mutation that caused a longer beak to form, that's a beneficial mutation. So mutations can be beneficial. Most of them are not going to be beneficial. So to bring you back to something we've already talked about before so you understand what we're going through today, we've already talked about protein synthesis. So how we go from a gene up here, all right, which is just a segment of DNA that codes for a protein. And we talked about how we can transcribe that to a messenger RNA. That messenger RNA is broken up into segments of 3 called codons, which code for an amino acid sequence down here. And that amino acid sequence makes up our protein. And so you can imagine any change that happens up here to our DNA. So if I was to just delete that C, that is going to affect that DNA. Everything's going to have to slide this way, and my DNA strand changed, my messenger RNA is going to change, my resulting amino acid sequence is going to change, the protein is going to change, and proteins control who we are. And so that would cause an effect. You could also imagine, what if this A got substituted and became a T? Well, that would affect the resulting process down here. This would no longer be a U, this would be an A, so on and so on, could affect our amino acid sequence. So the point being here is mutations occur at the DNA level, primarily when DNA is being replicated. There can be mutations that happen. And if that mutation happens to a sperm or an egg, and that sperm or egg is the sperm or egg that fertilizes together to make the new offspring, then that trait or mutation could be passed down to the next generation. Obviously, I can't just mutate and grow gills right here and start swimming. Mutations don't happen in individuals. They happen to gametes or sex cells and get passed down if those just happen to be the gametes or sex cells that fertilize to form the new offspring. So let's get started with a couple of our primary mutations that we have. There's 2 kinds. There's point mutations and frameshift. This first one here called point mutations. This is where we have a mutation that happens at a point, and it's because of a substitution, not a deletion of a nucleotide, not an addition or insertion of a nucleotide, because of a substitution. So if we look up here to the top, we have this messenger RNA strand. It's already been transcribed, and you can see here we know that each segment of 3 is a codon, and it's a segment that codes for an amino acid. So AUG codes for MET. And so each one of those codons puts together an amino acid sequence to produce our protein. So how can some of these affect it? So let's look at the first one. If we look right here, we have an amino acid sequence where this used to be a C up here at the top. So here's that C, but it got substituted with a U during the process of DNA replication. And so now rather than GGC, it is now GGU. But you'll notice this codon right here, this mutation did not affect this codon. It didn't affect this one, or even this one. It only affected this codon at this point. That's why we call them point mutations. And so, but you'll notice what amino acid did it code for? GGU coded for GLY, which is the same thing that GGC coded for. And so because of this mutation did not affect our amino acid sequence, we actually call it a silent mutation. So it's silent. It was a mutation, but it didn't get expressed in a different way. It actually expressed the same exact protein as the original GGC did. All right, so if we come down here, let me get rid of some of that. If we come down here to the next one, here's another mutation. And if you'll notice here, this used to be a G up at the top, and we substituted an A in its place. Or we didn't, but the mutation did. And so now this codon reads AGC, whereas before it read GGC. And this one actually caused for a different amino acid to be coded right there. So this changed our protein. We actually have a different amino acid sequence now than we did with the original protein that we should have had. And so what they call this type of point mutation, they call it a missense. All right, meaning that it's missing the correct amino acid. And so we call it missense. And so that's just a point where, again, that did affect our amino acid sequence, which in turn is affecting our protein. The biggest one that we can have there that can cause the most problems is one we call a nonsense. And there's a real easy way to remember this one. What happened here with this mutation is there was a change of this A over to a U. So this point was an A before, now it's a U. So this codon now reads UAG, which actually codes for our stop codon. And so when we have a point mutation that causes for a premature stop in the production of the protein, that's nonsense. All right, so we call it a nonsense mutation, point mutation. And so again, these mutations all happen because of a substitution. Sometimes they can have no effect to the protein, and we call it silent. Sometimes it could change the amino acid sequence, and we at that point, and so we call it a missense because it's missing the correct one. Sometimes it can completely cause the thing to stop the production of the protein altogether, and that's nonsense, or a nonsense mutation. All right. The next one we have is what we call a frameshift mutation. All right. So let me give you a clue of what a frame is. We have our DNA strand up here, and it's getting transcribed to this messenger RNA. And if you remember, I always taught you to go ahead and break those messenger RNA up into segments of 3, which we know are our codons. And each one of those codons codes for a particular amino acid. I want you to think of each one of these codons as a frame. And so if we were to delete a nucleotide, so now we're not talking about substitution, we're talking about deleting one out altogether, or insert one, it's going to cause all of those nucleotides to shift one way or the other. And so what we're going to get is a frame shift. And so think of each one of these as a frame. So UCU is a frame. It's a codon that is producing this amino acid, which is serine. All right. CAU, that's a codon, or that's a frame. GAU, and so on. So let's say we were to take this A and it gets deleted during the DNA replication process, which means that's also not going to be there. Well, what has to happen to all of these other nucleotides that occur after it? They're all going to slide this way, and you can see it's going to affect all of our codons because there's going to be a frame shift. And so if you come down here at the bottom, let's see how that affected it. It's already been deleted out for us down here. There's our first frame, our second, our third, and then here we don't even know what this one is right now. So, but here's the difference. You'll notice UCU is still the same as what we had here at the top. UCU, so it's still producing serine. We had CAU before right here, but because that A got deleted, so did the U, we had a shifting of all of these nucleotides this way, so now our codon here reads CAG, which is producing glutamine, which is totally different than the amino acid that should have been produced there. Same thing here. We have AUC, whereas before, if you look up here at the top, it should have been GAU. Because of this frame shift, that codon changed as well, and this one, so on and so on. And so you can see frame shifts, they affect the protein in a very vast way. All right, so this is going to occur because of a deletion or an insertion. We're going to see a shifting of the frames that occur, affecting every codon that happens after that mutation, and then affecting in turn every amino acid within the sequence of the production of that protein. And so that's called a frame shift. Last thing I want to show you is a mutation that occurs, or just a couple common mutations that occur with chromosomes. If you remember, we've talked about these before. This is called a karyotype. And so this is just showing the chromosomes for this particular individual. And you'll notice we have 46 total chromosomes, 23 pairs. And if you're looking at this person, remember, we can tell the sex of this person by looking down here at the X and the Y. This is a female. Why? Because she has 2 X chromosomes. For it to be a male, we would add an X and a Y. But this is a normal karyotype. This person has a normal looking karyotype. So we use this tool to try to identify chromosomal mutations. So let me show you an example of one where we have a mutation that occurred. You'll notice here at chromosome pair 21, we don't have 2, we actually have 3. And so we call this trisomy, tri meaning 3, trisomy 21. And there's actually 3 there, and this is actually a female as well, karyotype here. But what it does is it produces the trait or the syndrome called Down syndrome. And here's a picture of a guy with Down syndrome. I was doing a little research on, obviously, this screencast, and I came across this guy. A really cool story. He's had Down syndrome, obviously, his whole life, born with it. You can see his name there. Look it up. You look up his story if you want to. It's real interesting. He's a great power lifter. He's 32 years old, but he obviously has 1 extra chromosome 21. So he don't have 46 chromosomes. He actually has 47. And generally, there are some other syndromes where you can have 1 extra chromosome and it still produces a viable offspring here, such as John here. And so that's one example. Another example here, if you look here, I'll give you just a second. This is a very rare case where you can have actually 1 less chromosome somewhere. Have you identified where it's at yet? All right, there it is, right there at the X chromosome. This is actually a female karyotype. She should have 2 X chromosomes here, but she only has 1. This is a very rare case where you can have 1 less chromosome, 1 less chromosome and still survive. Generally, most of the time, if you don't have at least 46 chromosomes in that new zygote that forms from a sperm and an egg, that baby doesn't develop into a viable offspring. This is one case where it does, and they actually call this Turner syndrome. And I'll show you a picture here of somebody with Turner syndrome. This young girl, you can see here, one characteristic is a webbed neck. She actually had a surgery that got rid of that. But that's Turner syndrome. So that's just an example where you can have 1 less chromosome. So she only has 45 total chromosomes, and she still survived from that whole thing. So again, these were mutations, and mutations occur when there's changes in the DNA that affect the amino acid sequence of the proteins. So if there's a change in the protein, proteins control our physical traits. It's going to change the physical traits of us. So I'm Coach Boyson. Hopefully that was helpful to you guys. Have a good day.